So we stopped wherein we told that how we are going to encapsulate our somatic embryos, and I told you that hydrogels are comparatively better than the desiccated cells, uh, seeds. Okay, and also we saw that how hydrogels are being produced by using the sodium alginate and calcium chloride. Finally they form the calcium alginate beads, which is encapsulating our somatic embryos. Okay, so why that desiccated embryos, when you compare desiccated embryos and hydrated embryos, which is better? We told that it is the hydrated encapsules because they have a higher conversion frequency. And we also told that even APA can be added, right? Abscisic acid can be added, amino acids can be acid, your malic acid can be acid added, aspartate can be added, salts like potassium malate, potassium aspartate, these are all can be used. Why are they used? What did I tell? Why are they used? Because to increase the vigorness of a synthetic seed. Because we told that whatever the synthetic seed is, compared to the normal seed, these are less vigored. Okay, that is the viability is also comparatively less. The vigorness is less. Environmental protection is less. So we wanted to make our synthetic seed as equal as the natural seed. Maximum we are trying. And that is why we are using all this. That is why we are using abscisic acid, right? To improve the development of the embryo, the four stages what we spoke. And we saw that the oxygen is a limiting factor. So what did we use? We used hydrocarbon, right? Because they have a higher solubility of oxygen in comparison to the water. So you add a hydrocarbon, it will have what? Higher solubility, it will dissolve the oxygen, and then it can be taken up by the seeds. So somatic embryogenesis or synthetic seeds can be used in the germplasm conservation. So we have dealt with this. See, this is before I go into the application part, just will recall what establishment of callus culture. Okay, for artificial seed, you can you can go for a callus culture, or you can produce even somatic embryogenesis also by direct or indirect method. Then what you do, maturization of somatic embryos should occur, all that four phases. Okay, then you encapsulate your somatic embryogenesis as we dealt in last class. After encapsulation, the artificial seeds are tested by two steps. Test for embryoid to plant conversion. See that whether your embryoid are getting converted into the plant. That is the first and foremost you have to see that. Okay, they are converting into plant. Then take them to the greenhouse and fielding planting. That is, in the greenhouse you provide all the environmental conditions which is as similar to the natural environment, and see whether that plant is growing. These two testing should be done, and only when they pass these screening tests you can go for the what commercialization. Okay, until unless that any of your tissue culture without the field trial, you cannot go for commercialization. You cannot sell it to the farmer or anything. Okay, so so this was the synthetic seeds. Look at this, how beautifully they are growing into the uh, new plant. Okay, bipolar I told, no, shoot and roots will be developed. This one also we saw. Now we shall look into the application part. See, to get a true seed, we have to wait till reproduction phase is complete. Here, what do you mean by true seed? In natural environment, okay? If I want a seed, I have to wait. It has to fruit, and then I'll get the seed. It is seasonable dependent. Some will take years together to obtain a seed. So we have to wait till the reproduction phase is complete. But artificial seeds are available within at least 1 month. Okay, so that this is really amazing. So plant bear flower and produce seeds at particular season of a year. But the production of artificial seed is not time or season bounded. We need not wait for that particular season, for that particular climatic condition to come. So since everything is in under our control, our artificial seeds need not wait for a particular time or some particular season. We will provide you everything. That's time, season, everything we shall provide you. The work on some plants is delayed due to the presence of long dormancy period of their seeds. Particularly, you can see that this woody trees, how long they take for the production of the seeds. So many years they take, right? So by growing the artificial seed, this period may be reduced. So these, look at the wonderful um, positive effectiveness they are having. Artificial seed coating also has a potential to hold and deliver the beneficial adjuvants such as growth promoting thizobacteria, plant nutrients, and growth control agents and pesticides. What this means, see, okay, I will go back to the picture and I'll, okay, this is your seed, right? And it is encapsulated. Now, what can I do? Inside this, I can add nutrient. I can add thiobacteria. What is the thiobacteria? It produces certain growth regulators. Okay, growth promoters it produces. So I can use that. I can add nutrients into it. So along with the encapsulation, along with my somatic embryos, I can add all this adjuvants such as the growth promoting your thizobacteria. I can add my plant nutrient. I can add certain agents which can control the pest and then grow the plant. So, artificial seeds help to study the role of endosperm and seed coat formation in the in vitro cultures. This is another important point wherein where the researchers are doing a lot of study how what is the role of endosperm, what is the role of the seed coat. This all can be studied in vitro. Instead of going to your field, you can study them in the laboratory itself. And the best thing, small amount of space is required. Okay, because you saw that in a small space, we are growing so much of plantlets, all in glass bottles, tissue culture bottles. So you can conserve and store large amount of plant material. Then it overcomes the problem of climatic dependency or environmental dependency. That is what happens. Those plants seeds which are growing in the environmental condition, they are dependent on the climatic conditions. I told you, no, like tropical plants cannot grow in temperate cannot grow in tropical or in subtropical. It becomes difficult. And also they are dependent on the environment conditions. Okay, sometimes because of the natural disasters which occur, the plants are put into what lot of stress. I mean, they're washed away. So maybe there is flooding, there is drought, everything happens. Okay, Then it provides large amount of material for culturing for regeneration of the regeneration later. So such type of climatic dependency is not there in the in vitro, because it is growing in the laboratory. You are not going to create any drought or any or what to say, cold conditions or whatever which is unfavorable. You are not going to do it unless until that particular experiment you want to see the stress, study the stress on the plant. Only then you induce high salt concentration, whether it can withstand a particular drought condition, whether it will withstand a higher temperature. When your point of study is that, then you're doing this. Otherwise, you will not do all those things. Okay. Then cryopreservation. You can preserve also your all this germplasm. I told you in the gene bank. So this all this somatic embryogenesis of synthetic seeds can also be used for the germplasma conservation. Okay. See, you can do HC2 conservation also, wherein you grow the plants. Okay. There are so many government laboratories wherein they grow, or even the private laboratories wherein they grow, and see to that they are not harmed. But unfortunately, they have to face the such type of climatic dependency or environmental dependency. So in order to avoid all this, you can keep it in the gene bank itself. Okay. So generally the seeds used for germplasm conservation, you provide suitable condition in the gene bank to preserve this material. So what you do, you preserve them either in the form of seeds or synthetic seeds or different forms of in vitro cultures, like shoot meristem, uh, or you can store it in the form of a shoot or in the form of a meristem. Okay, but only thing what is the thing is, you have to provide them the nutrient so that they survive for the long period of time, and you have to do some subculturing and all. Otherwise, you can also cryopreserve them. So you know cryopreservation at minus 196 degrees centigrade for the liquid nitrogen. But you should be careful whether that plant will really going to withstand the sub-zero conditions. You have to keep that in the mind. And cryopreservation, the viability is not lost during the prolonged storage period. And why? Why is that viability not lost when you keep it at one minus 196 degrees centigrade? Even your animal cells, you're keeping it in minus 90 196. Plant cells you're keeping, microbial cells also you learned that you're keeping at by 196 20 degree, sorry, 196 degrees centigrade in liquid nitrogen. That is called as cryopreservation. Why we are keeping that? What is happening? So here, what happens is that the you are actually, the activity of the cells will become a standstill, static. Okay, they can be arrested. So that is the reason the metabolic activity, that is, they are not dead, they are alive. Only they are metabolically inactive. You studied this clearly in your microbiology also. So this is one of the positive thing, that is you cryopreserve them, and then you can use them whenever you wanted, because you are arresting the cell division. But there is also a problem in cryopreservation. What is the demerit? The cell's viability is important, we told, right? The vigorness and viability is important, and that can be lost when you are keeping it at such a low temperature. Water in the cells freezes, so there will be crystallization happening. It will form crystals. Okay, see, ice crystal formation occurs when you're keeping in such a low temperature. And then what they do, they will directly damage the membranes. of your seeds, encapsulated seeds, they will directly damage who? Your crystals. From where the crystals are formed? From this liquid nitrogen. Because of that cold condition, the crystals, water crystals are being produced and they will damage your seeds. Okay? They are going to damage your seed which you have stored. So you're storing to see that they don't lose their vigorness, they don't lose their viability. But what happens? The same thing which you don't want to happen, that is going to happen here. So what do you use? You use cryoprotectants. Now in nature also you see plants grow in the snowy region, right? Very low temperature also they are growing, right? So, what is happening there? There must be some factor in that plant which is protecting that plant from that high, with that low, very low temperature. That is, that plant has learned to acclimatize itself to that natural environment. So, if it has to get acclimatized to that environment, that low temperature natural environment, some biophysical changes is happening in its body, or biochemical changes is happening in the body, right? So scientists have studied that. How come there are certain fishes, certain animals which are living in the icy conditions, certain plants are surviving in this condition, in the cold condition? What are the factors which is making them to withstand this low temperature? And they have come to a conclusion saying that okay, they are cryoprotected, they are getting acclimatized to that condition, that cold condition. They are cryoprotections. So similarly they have developed cryoprotection to these uh, synthetic seeds also, same as in the nature. That is, the antifreeze protein are being produced in this those plants which are growing at a very low temperature. So, like polyalcohols, proteins which can act as a substances which can act as a osmoticums. Okay, they start producing in those plants because of that, like these polyalcohols or osmoticums. Because of them, they are able to withstand that, and certain biochemical reactions which is happening, down regulation and up regulation of the genes which is happening, they are able to withstand. So what it our scientists did, they understood that phenomena and they started adding the psychro cryoprotectants. And what is the commonly used cryoprotectants? You have studied this also in your microglycerol, right? We store a microorganism where? In glycerol. For we can keep it for years. Okay. So you prepare a stock of glycerol and you can use it. Okay. Some plants require different concentration, another plant require another different concentration of glycerol. So that stock depending on the species you are working, whether you want to cryopreserve your plant or your bacteria or animal cells. Based on that, various glycerol concentration have been utilized. So, and there is also like this dimethyl sulfoxide is also being used along with this DMSO. You must have heard this also. DMSO is also being used for your cryopreservation. So these all are called as cryoprotectants. Depending on the cryoprotection is necessary for, I mean, your cryoprotection is necessary for cryopreservation of the plant material unless they are naturally dehydrated. So generally, when we say this cold acclimatization in plant, it is nothing but a slow dehydration process. So these are the important applications, okay, of the synthetic seeds. How does we studied like how the synthetic seeds are being produced, and what is the final form, and what all is required, what are its application. We have looked into it. And lastly, let us also look into the protocol of developing this synthetic the somatic embryos for the production of synthetic seeds. The protocol is usually you can take for your somatic embryogenesis, you can take a leaf petiole or root segments from 7-day old seedling or cambium tissue. From that leaf, or you're taking cambium tissue or the root segments. Okay. So what are these called as? These are called as explants, and they are placed individually on the semi-solid morassic shoe media. So this is what is my explant. And where did I place my explant? I placed it in a semi-solid media, that is the morassic media. Where did you place? I placed it in the culture tubes and incubated in the dark. Now, why in the dark? Because particularly when I keep this media or the explant in the dark, it develops into a callus tissue. Okay. Next, after 4 weeks of callus growth, what you do? You're going for cell suspension culture. I told you callus will become friable. You have to agitate it, put it in the rotor shaker to get suspension culture. Only then you get a single cell culture. So callus, what did I get? I got the suspension culture. So after 4 weeks of callus growth, cell suspension culture is to be initiated. How? By transferring 0.2 gram of the callus to the 250 ml of the Erlenmeyer flask. That is like your conical flask containing 20 to 25 ml of liquid media of the same composition. And it is shaken on a rotary shaker with 125 to 160 rpm at 25 degree centigrade. So you grow your callus. After that, you grow your suspension culture. Always I told you that suspension culture, you have to use a rotary shaker. Okay. So what is the use of this rotary shaker? That is like you get single cells. Okay, the cells can be broken up easily so that you get a single cell. Not only that, there is a what easy movement of the airflow, okay, in the flask. So you maintain the particular rpm and the temperature. After that, what you have to do, the cell suspension are subcultured for every 4 weeks. We should go for subculturing, otherwise there will be no nutrition, desiccation will occur, and ultimately your experiment is dead. Cell suspension are subcultured for every 4 weeks by transferring 5 ml to 65 ml of fresh liquid media. So 5 ml to 65 ml of fresh media you add for every 4 weeks so that they keep on, you can retrieve your cells, keep them alive. Then induce a uniform embryo population. Cell suspension is passed through a series of stainless steel mesh sieves. Why? Because you have to get uniform embryo, all of same size. Synchronization is important. Okay. To induce somatic embryogenesis, portion of your sieved cells, now you all got your sieved cells up there. Now what you have to do, you have to induce embryogenesis. So what you're going to do, you transfer it to the 2,4-D, are transferred to 2,4-D free liquid media, or cell suspension can be planted into a semi-solid MS media devoid of 2,4-D. So, induction, whenever you're going to induce, you're going to add the auxins. One important auxin is your 2,4-D. Or otherwise, if you are going to, if your cell suspension are going to be planted in semi-solid MS media, no need of adding your 2,4-D. Okay. For normal embryo development and to inhibit the germination, especially elongation, ABA, your abscisic acid can be added to the culture media because this is the one which is involving in the development of the embryos, all the four stages of their embryo. Cultures are incubated in dark. After 3 to 4 weeks, somatic embryos can be placed in the agar medium devoid of 2,4-D for the plantlet to develop. That is, you remove your auxins and transfer it to your cytokinin. I told you that benzylaminopurin, indole acetic acid, and gibberellic acid combination is very good. I told you, and and also high sucrose level, abscisic acid. All these are essential for your what maturation, initiation, maturation, development, germination, development, all this. Okay, then once it has been developed, so your somatic embryos, plantlets are finally transferred to the pots or vermiculture, that is the compost, okay, for subsequent development into a whole plant. The adventitious embryo is a bipolar structure that develops into a complete plantlet, and there is no need for separate root phase as with the shoot culture. That is usually what they mean to tell is, whenever you studied about the other cultures, what did we tell? We first produce the root, then we remove the uh, um, what to say auxin, and then we add cytokinin so that let the root start developing. Here that thing is not there because it is a bipolar structure. You will get your radical and a pumule formation, and this is how you produce your a protocol for production of your somatic embryogenesis. A quick recap: take the explant from the 7-day old seedling, place the explant in the semi solid morassic media, culture them in the dark. They will develop into the callus tissue, and after 4 weeks of their callus growth, grow them as a suspension culture in an Erlenmeyer flask using a rotary shaker. Obviously, it is a liquid medium. Okay, suspension when the word comes, and you use for 125 to 160 rpm at 25 degrees centigrade. Cell suspension are self-cultured for every 4 weeks by transferring to 5 to 65 ml of fresh liquid media so that we can sustain and retain the viability of the cells. After that, what you do is all your single cells, they should be in a uniform for the uniform embryo production or synchronous cell division. You sieve it in the steel mesh. Okay, you'll get all the, because of the seeds will be of uniform size, you'll get a uniform cells. After that, you add it into 2,4-D liquid media. Otherwise, if you're using semi-solid MS media, no, not necessary that you have to add 2,4-D. And then what you do for normal embryo development, okay, and to inhibit precocious germination, that is, I told you, no, they germinate very fast. When they germinate very fast, what happens? The seeds are very weak. So to avoid that, and especially the root inoculation, we add the abscisic acid can be added into the culture, and the cultures are incubated in the dark. After 3 to 4 weeks, somatic embryo can be placed in an agar medium which is devoid of 2,4-D for the plantlet development. So you use your cytokinin, all those things for the maturation and all the four stages of your, what is that, globular, heart, torpedo, cotyledonary stage. It passes through all the stage, which is very similar to your zygotic embryogenesis. Finally, you put it into the pots, sterile soil containing your compost, and develop the plant. And it is a bipolar in structure, so there is no question that you should put it into shooting media, rooting media, nothing like that. Shoot and root is being produced. And this is from a journal I have taken, how they have developed a coffee somatic embryogenesis. Okay, look at this. This is the mother tree. They've taken the explant, and embryogenic callus is being developed. Cell multiplication occurs. That is the progressive division of the cell. That is the embryogenic clumps. What did I tell you? That is being produced. Okay, the embryogenic clumps will be produced. That is before the formation of the embryo. genic cell, embryogenic clumps are formed, and then embryo cells are produced, and they repeatedly undergo division. Okay, and finally see that they are all putting it into the bioreactors to propagate more number, and then they do the field testing, direct sowing, and plant acclimatization in ex vitro. And these are the various steps. So with that, we come to the end of somatic embryogenesis. So we studied about what the definition of somatic embryogenesis, how the somatic, what are the conditions, what is required for the somatic embryogenesis to produce, that is the direct and indirect method. And we told it is a bipolar, and want to go through the maturation, induction, germination, developmental stage. How should I change the growth regulators? We studied, and we studied the four stages of the embryo development. Okay, that is up to the shoot and the root formation, and then we see that how they react in the greenhouse, and when they have passed the screening test, we bring it to the field. Afterwards, we told that since they can be used for clonal propagation or for somoclonal variation, if you wanted to transport to other regions, or if you want to want to add more nutrients or add pesticides into it, you can go for encapsulation. And encapsulation, I told you can go for desiccation or hydrogel. Desiccation is drying to 8 to 15%. If your seed is not unable to withstand desiccation, you can go for hydrogels, and particularly hydrogels have shown better conservation and better frequency of development. And apart from them, we saw the what is the applications, various applications of the synthetic seed, how synthetic seed is produced. Also we saw using the sodium alginate and calcium chloride, they form the calcium alginate beads. And then we saw that various type of aminos and polyamines or organic acids are added, abscisic acid is added to see that they are the somatic embryos retain the vigorness, viability. Okay, and grow into the required uh, plant. And we also saw that how can we preserve these in germplasm. We can even preserve nowadays in cryopreservation, but you can add a cryoprotectant. Okay, like your glycerols or DMSO can be added. So I think so there is a four set of videos which we have made on somatic embryogenesis. You can go through all. Okay, I'll be numbering it. Okay. With that, we conclude our somatic embryogenesis class.